Hello students, welcome to my channel MADHU'S Globe. Today we're gonna look into heme catabolism, or you can say degradation. Catabolism or degradation, both are same. So let's begin without any further delay. So we all know what is heme, and this heme is going to be catabolized or degraded, which means heme is going to be degraded into pieces. So as we all know, where we can see the part heme or the compound heme, actually we all know there is an RBC, red blood cell. It transports oxygen and CO2, oxygen to the tissues, CO2 to the lungs. So this is the function of RBC. Now every cell in our body has a particular lifespan. Similarly, RBC also having a particular lifespan, which is 120 days. Okay, 120 days. After the lifespan of RBC is completed, 120 days is completed. Actually, RBC, where can you see the RBC? In the circulation. If you can see the blood vessel, see here, let me change the color so that, yeah, this is a blood vessel and these are RBCs. These are RBCs. Once the lifespan of these RBCs, 120 days, are completed, what happens? These RBCs will be transported to the reticuloendothelial system of spleen, liver, and some other reticuloendothelial systems of other organs. So mainly spleen and liver will be involved in heme formation and degradation of the same heme. As we can see, once 120 days of RBC is completed, what happens to the RBC? What after 120 days? This RBC will be removed from the circulation. See here, this RBC will be removed from the circulation and it will be entered into the spleen. So here is the spleen, it entered. Inside the spleen there are macrophages. So macrophages, they engulf this expired RBC. After 120 days it will be expired. This expired RBC will be engulfed by the spleen macrophages, and inside the macrophages the process will be initiated. What happens? The first process of heme degradation. As we all know, inside there is a heme. The heme structure looks like this. So four porphyrin rings, as you can see here while I am drawing. So 4 pyrrole rings and connected by methylene bridges like this, and inside an iron atom Fe and iron atom Fe2+. So this is a heme structure. Heme will be connected with or bound form with the protein. See here, these are all proteins. Let me change the color so that you can appreciate everything clearly. Okay, this is all these 4 are porphyrin rings, and to it a protein is added. This is the one protein. Okay, now porphyrin ring 1, 2, 3, 4. 4 porphyrin rings will be the sites of oxygen binding. 1 hemoglobin, 4 oxygen molecules will be, they have 4 sites for oxygen. So this is how the hemoglobin is arranged. HB, which means hemoglobin. So here we have 2 parts. Globin part, which is a protein part, 4 globin chains, alpha 2, beta 2. In case of adult hemoglobin, globin chains are alpha 2, beta 2, 2 alpha chains, 2 beta chains. Here 1, 2, 3, 4 protein chains and 4 pyrrole rings. So this exact structure will be incorporated once this hemoglobin is synthesized during the synthesis of RBC. This hemoglobin will be incorporated into the RBCs. See here, this is a hemoglobin that we had drawn here. Okay, porphyrin rings are there, methylene bridges, inside Fe2+. So this is how the RBC is made. What happens after 120 days? After 120 days, what happens? That is the point that we are going to discuss on. Yeah, this is the RBC inside hemoglobin. 120 days are completed. Now this RBC will be engulfed by the spleen, and inside the spleen this RBC membrane will be broken down, and inside hemoglobin will come out. Okay, inside hemoglobin will come out. So what happens next, we'll see in this chart. That I had captured this image from U. Satyanarayana biochemistry book. I also advise you to take the same, follow the same book for the preparation. After watching this video, you can go through U. Satyanarayana book for this chart. So U. Satyanarayana book chart is very easy as compared with other books. So I advise you to go through this U. Satyanarayana book, heme degradation chart. So here I already told you age erythrocytes after 120 days. Let me change the color, a bright one. Yeah. Aged erythrocyte after 120 days of RBC, they are aged erythrocytes. We all know, right? So they will be engulfed by the spleen macrophage. Here see, spleen macrophage. Now RBC membrane broken down, and hemoglobin is extracted. Now hemoglobin is here. This hemoglobin will, I told you hemoglobin means two parts, heme plus globin, hemoglobin, which means heme one part and globin second part. Hemoglobin will be separated from, heme will be separated from globin part. See here, these are globin parts, protein chains, alpha 1, alpha 2, beta 1, beta 2. These are separated, only heme is remain here. The remaining heme, actually hemoglobin is not only the source for heme, some other compounds in our body called cytochromes, non-heme protein, cytochromes also the sources of heme. So we get 80% heme from hemoglobin. Hemoglobin supplies 80% heme, and other non-heme proteins like cytochromes and all, they provide 20% heme. So totally 100% heme is here. Ultimately the heme undergo degradation. So degradation is started already in a macrophage. Heme is produced, the globin parts are separated, beta, alpha, all these separated. These pink colored dots are called hemes, porphyrin rings. See here, heme is there. So these proteins will be degraded into amino acids, and these amino acids will be reutilized for other protein synthesis in the body. Now this heme, once it is produced from hemoglobin, this heme will be converted to biliverdin. So by the enzyme heme oxygenase, very very important, this heme oxygenase. So here what happens, as I already told you, this is a structure. This is a structure of heme. This is structure of heme connected by methylene bridges, and inside Fe2 plus is there. Once heme is broken down into biliverdin, so like what happens, heme, like one methylene bridge will be what happens is heme oxygenase, by the action of this heme oxygenase, this circular structure will get linear, which means the bond will be broken. So what happens once bond will be broken, this iron atom will come out. See here, after heme acted by this heme oxygenase, heme will be degraded into biliverdin, and inside Fe2 plus will come out in the form of Fe3 plus by this heme oxygenase, and also one carbon monoxide will be released. And here this is a energy dependent, which means NADPH plus H plus is required, and one oxygen is also required. So by using NADPH plus H plus and by using NADPH plus H plus, heme is converted to biliverdin by the enzyme heme oxygenase, and Fe3 plus and CO2 both are released. Now this biliverdin again will be reacted with the enzyme biliverdin reductase. Biliverdin reductase catalyzes the conversion of biliverdin to bilirubin. Now the bilirubin is formed. So what is bilirubin? It's a compound produced from heme. It's a pigment produced from the heme in macrophage. See here, from hemoglobin to, right from hemoglobin to bilirubin production took place inside the macrophage of the spleen itself, or reticuloendothelial system, we can say macrophage. This is all happened up to here, happened in the macrophage. After bilirubin is formed, you can also see here, I'm just clearing them off, all these scribblings, so that you can easily see that. Now after bilirubin is formed in the macrophage, this bilirubin is insoluble. Of course, whatever we are getting from hemoglobin after the lifespan is completed, all these degradable products are waste products. They have to go out from our body. So we have to wash them off from our body. Otherwise, these waste products, bilirubin, bilirubin, all these waste products will get accumulated in the blood, and it leads to toxic effects to the brain. Okay, so we have to eliminate this expired heme in the form of bilirubin. But here the bilirubin process has to go a further organs to get solubilized. See here, bilirubin is formed, which is insoluble form in the macrophage. It has to go other organs also to convert into soluble. Actually, insoluble bilirubin, it cannot be solubilized in a blood. That's why bilirubin takes help from albumin. Okay, albumin is a protein present in a plasma, blood plasma. This albumin carries this bilirubin to the liver for conjugation purposes. As I already told you, bilirubin, since it is insoluble, it cannot be circulated in the blood. Hence, bilirubin takes help from albumin. Albumin takes this bilirubin up and takes them into the liver. So bilirubin will be taken up by the albumin into the liver. Now this bilirubin, formed bilirubin in the macrophage is present in the liver. So liver is a major site for solubilizing the bilirubin into insoluble bilirubin into soluble bilirubin. So for the soluble for conjugation, you can say soluble bilirubin is like insoluble bilirubin is nothing but unconjugated bilirubin. Let me arise and write unconjugated and conjugated terms. Bilirubin here is insoluble, as I already told you in a previous step. Insoluble is also nothing but unconjugated bilirubin. This unconjugated bilirubin has to convert conjugated bilirubin. Okay, unconjugated has to be converted to conjugated bilirubin. So this conjugated bilirubin is nothing but soluble. So this soluble bilirubin can be excreted. Okay, now this bilirubin in the liver will be converted to bilirubin diglucuronide. This is a soluble form. So this reaction is done by bilirubin glucuronide transferase, which is a very, very important enzyme in the liver. So this enzyme in infants and newborns will be inactive or sometimes not fully functional. So what happens? Bilirubin cannot be converted to bilirubin diglucuronide. For this reaction, 2-UDP glucuronate is required. This 2-UDP glucuronate will be combined with this insoluble bilirubin and converts this insoluble bilirubin into soluble bilirubin in the form of bilirubin diglucuronide. These two glucuronides will be attached to the bilirubin and this bilirubin will get solubilized in the circulation. Now, once liver conjugated the bilirubin into bilirubin diglucuronide, which is soluble form, insoluble into soluble form, this soluble form of bilirubin will get entered into the gallbladder. Like after the liver, what you can see, this is a liver and here there is a gallbladder. So once here is a bilirubin which is unconjugated form, which is insoluble form, will be converted to conjugated bilirubin. This conjugated is soluble now, it is ready to go out and excreted. So now this conjugated bilirubin enters into the gallbladder. So here this is a gall duct. As we all know, this is a bile duct. Okay, this is a bile duct. Through this duct, this conjugated bilirubin will enter into the gallbladder, and from here there is one more connection from a bile duct to the intestine. So here exactly the ampulla of Vater. So this conjugated bilirubin comes through this bile duct and enters into the duodenum. From duodenum, what happens here? Once bilirubin diglucuronide is formed, this will get enter into the gallbladder along with bile salts. Now, bilirubin once enters into the duodenum and followed by the large intestine. Okay, followed by the large intestine. So here what happens, inside the large intestine there are microbial flora, bacteria, useful bacteria, this synthesize vitamins and all. So this bacteria utilizes this diglucuronide, bilirubin diglucuronide, which is soluble form, and convert them into urobilinogen. So intestine and kidney. So here what happens, this bilirubin diglucuronide enters into the intestine, and by the microbial enzymes in the intestine, they convert bilirubin diglucuronide into urobilinogen. This formed urobilinogen will be entered into the blood circulation again. So in the blood what happens, urobilinogen enters into the blood and they'll go to the kidney. So urobilinogen enters into the kidney, and in the kidney urobilinogen is converted to urobilin. So when urobilinogen is excreting by the kidneys, it is called as urobilin, and it will be excreted into the urine. So now urobilinogen to urobilin will be excreted into the urine. So this process happens in the kidney. In case of feces, what happens? Urobilinogen will be converted to stercobilin by the microbial enzymes. These microbial enzymes convert again urobilinogen to stercobilin. This stercobilin will be entered into the feces and will be eliminated. Here excreted, here eliminated. Okay, so the characteristic brown color of the feces is due to stercobilin, and here deep amber or yellow color of urine due to urobilin. So since urobilin is present in urine, we are calling the we pronounce the color of urine as yellow in color, and also stercobilin, due to presence of stercobilin in feces, we are saying the color of feces is brown. Characteristic feature of stool color is brown due to stercobilin and urobilin. So this is a degradation of heme or catabolism of heme, all about heme catabolism and degradation. What happens, I'm just recall everything again. What happens here, after 120 days of RBC lifespan, heme will come out, whole RBC will be engulfed by the macrophages in the spleen. Then the macrophages, they separate hemoglobin from RBC. This hemoglobin will be splitted into two parts, heme plus globin. Heme is here and globin parts are here. Heme is here. Two parts are separate from hemoglobin, and this heme will be converted to biliverdin. Biliverdin will be converted to bilirubin. Now this bilirubin, this whole process happened inside the macrophages, and this bilirubin will transported to the liver for further conjugation purpose, since bilirubin is insoluble, and this bilirubin, since it is insoluble, it will be taken up by the albumin in a blood. So throughout the blood circulation, albumin carries the bilirubin and transports the bilirubin into the liver. In the liver, bilirubin will be converted to bilirubin diglucuronide, which is a soluble form of bilirubin, and it can be easily excretable and eliminated. So here bilirubin diglucuronide will be entered into the intestine, and by the intestinal microbial enzymes, it will be converted to urobilinogen. This urobilinogen in the kidney will be converted to urobilin, and this urobilin will be excreted finally into the urine. And this urobilinogen, same urobilinogen, further action by the microbial enzymes will be converted to stercobilin. This stercobilin will be eliminated by the feces. So due to the presence of the stercobilin in feces and urobilin gives them color. Okay. So now this is all about heme catabolism and degradation. If you like this video, please hit the like button, subscribe it, and if you have any queries, drop them in a comment box. Thank you.